Welcome to our third video for Chapter 4. Now we're going to focus on non-proteobacteria as well as phototrophic bacteria. In this section, we're going to go over the unique features of each category, the non-proteobacteria as well as the phototrophic bacteria. And we're also going to identify and give several examples of each type or each group. Most gram-negative bacteria fall under proteobacteria. Non-proteobacteria are the other gram-negative groups outside of that phylum. And we're going to look at four classes of non-proteobacteria, including chlamydia, spirochetes, the CFB group, and planktomycetes. At the end of the section 4.3, we're going to also look at a different group known as phototrophic bacteria. These are bacteria that can use light to produce energy, and this group will include both proteo and non-proteobacteria. Chlamydia are obligate intracellular pathogens, and this means they must live inside a host cell. In this case, it's because they can't make their own ATP. Their life cycle alternates between two forms. We have elementary bodies, which are inactive but infectious, and reticulate bodies, which are metabolically active and they replicate. So unfortunately, chlamydia are resistant to many of our host defenses, like the immune system, and they can spread easily. In terms of disease, chlamydia trachomatis causes trachoma, which is a leading cause of blindness. It also causes the sexually transmitted infection, LGV, or lymphogranuloma venereum. And in LGV, you see signs like genital blisters or swollen, painful lymph nodes in your groin, pelvis, or rectum. These infections spread very easily, so they're contagious, and this is why they're very common on college campuses. Here we see the life cycle of chlamydia, starting with those inactive elementary bodies that enter the host cell and convert to active reticulate bodies, which will multiply and differentiate into, again, those elementary bodies. Eventually, the bacterium uses up all of the host cell ATP, the host cell dies, and those elementary bodies are released. And elementary bodies are kind of like endospores. They're not endospores, but they're similar in that they're also dormant and hardy. They are able to survive harsh conditions, making it difficult for our immune systems to fight chlamydia. Spirochetes are long, thin, spiral-shaped bacteria, and they're so thin that they're hard to see with a gram stain, so we actually use dark field microscopy usually to visualize spirochetes. Spirochetes have a really interesting structure that is flagella-like, but what you actually have for spirochetes are endoflagella. These are axial filaments, and I'll show you a picture soon, that are found in between the regular plasma membrane and the outer membrane. Remember, gram-negative bacteria have their plasma membrane, a thin layer of peptidoglycan, and then their outer membrane. So these filaments are between those two membranes in an area called the periplasm. And when these axial filaments move, they give the bacteria a corkscrew motion. Examples of spirochetes that cause disease in humans include Treponema pallidum pallidum. This is the subspecies or the strain, and this causes the sexually transmitted infection syphilis. And another important one is Borrelia burgdorferi, which causes Lyme disease transmitted by ticks. We see spirochetes here in the dark field microscope. Over here we can see a cartoon depiction with the inner membrane, that inner line, the outer membrane, and the periplasm in between the two regions. And here are the endoflagella. So we see little dots that actually run the length of the bacterium, and bundles of endoflagella form that axial filament, which runs the length and gives the bacteria that twisting motion or corkscrew movement. Here's another look at the same thing. I like this one because it has different color coding. Red for cell membrane, so the regular plasma membrane. I see the peptidoglycan in gray, and the outer membrane in that kind of orangish brown layer, that outermost layer, and the axial filament in between the two in that periplasmic space or periplasm. For our spirochete checkpoint, remember that we usually use dark field microscopy to see spirochetes because they're so thin, right? They're very thin, long, spiral-shaped bacteria, making them difficult or maybe even impossible to see clearly with a conventional gram-stained bright field microscope. The next group is the CFB group, and this includes Cytophaga, Fusobacterium, and Bacteroides. These are rod-shaped anaerobes that are found in places like the gums of our teeth, that surround our teeth, the gut, so the intestines, or the rumen of ruminants, like cows. So the rumen is a compartment of the stomach that is important for fermentation in these animals. So fermentation is important because certain animals cannot digest polysaccharides, complex carbohydrates like cellulose. So these bacteria can break cellulose down into something that the animal can harvest energy from and absorb. For these bacteria, Cytophaga are aquatic bacteria that can glide, so they can move around and they live in aquatic environments. For Fusobacterium, they live in the mouth, but they can cause periodontitis and ulcers. So this is gum disease and ulcers. And then Bacteroides make up about 30% of the human large intestine microbiome. So that's a huge fraction of the bacteria in our large intestine. And they're usually mutualistic, so they're usually good for us, but sometimes they're opportunistic pathogens. And what that means is these are microbes that normally don't cause disease in healthy individuals, but these bacteria can take advantage of weakened immune systems or other underlying conditions to cause infection. For the CFB checkpoint, Cytophaga, Fusobacterium, and Bacteroides are in this group because even though they have different phylogenies, they're phylogenetically different or diverse, they do share similarities in their DNA sequence, in their DNA nucleotide sequences. They're also all gram-negative, they're all rod-shaped, they're anaerobic, and they're adapted to those environments I mentioned, the gut, the gums, and the rumen, and they play key roles as fermenters. Our next group are the planktomycetes. So these are found in freshwater, saltwater, or brackish water, which is a mix of fresh and salt water. And you see these or this type of water in something like an estuary. So these are interesting and unusual aquatic bacteria because they reproduce by budding. And what happens in this case is there's going to be a mother cell that will produce a smaller swarmer cell that detaches from the mother cell eventually. The swarmer cells are going to be motile. They're going to be able to move around, they're free living, but the sessile cells, the original cells, will be immobile. These will attach to surfaces with a structure called the holdfast, and these cells can reproduce. Examples of planktomycetes include bacteria that can recycle different environmental compounds in these aquatic ecosystems. Here's an image of those planktomycetes. We see a sessile planktomycete here on the left with a holdfast that keeps it in place. Imagine you're on the beach and you're letting the waves run over your legs. If you didn't have anything to hold you there, you would just wash away with the water. So the holdfast keeps them still, right? Lets them stick to a surface. Swarmers, though, are motile, and they do not have a holdfast. Table 4.7 from our book shows us the most clinically relevant genera of Nonproteobacteria, and I've bolded some of the most common ones, some that you're going to encounter in this course and in future courses, including chlamydia, which causes the sexually transmitted infection chlamydia, as well as pneumonia or trachoma, that blindness. Bacteroides, which is very common in our GI tract, Borrelia, which causes Lyme disease, and Treponema, of which Treponema pallidum pallidum, we said causes the sexually transmitted infection syphilis. Here's our checkpoint on planktomycetes. They are really interesting in how they reproduce because they reproduce through budding, right? So they bud. And I can see a larger mother cell, the original cell, and then the cell that is budding off. And this is different from most other bacteria because bacteria usually divide through binary fission, right? So they usually have, don't worry about the details here, but usually you have a cell that elongates, gets bigger, right? And it's going to divide in the center to produce two equal-sized cells. But in planktomycetes, we have the budding off of the mother cell. At the end of section 4.3, you'll see phototrophic bacteria. So now we're jumping away from non-proteobacteria and talking about this group of bacteria that can undergo photosynthesis, right? They can use sunlight as their primary energy source and use that energy to use sunlight to produce ATP. So phototrophic bacteria, in this group, we have both proteobacteria and non-proteobacteria. And the type of photosynthesis they go through can be oxygenic or anoxygenic, which means they do not produce oxygen. So oxygenic photosynthesis is what you typically see in courses before this course. So maybe in general biology, you saw this equation for oxygenic photosynthesis where they take carbon dioxide, so plants take CO2 and water and light and produce sugars, oxygen, and water, right? So this is oxygenic photosynthesis because oxygen is a product of photosynthesis. In anoxygenic photosynthesis, that's down here, you're not using water as your electron donor, and you don't form oxygen, right? So look, there's no oxygen here. So anoxygenic photosynthesis, you're not producing oxygen, and this is because instead of water, you're using electron donors such as hydrogen sulfide. Most of these phototrophic bacteria have pigments such as bacteriochlorophylls and carotenoids. So these are similar to chlorophylls, but not the same exact as plant chlorophylls. The exception is cyanobacteria, which I showed in the previous slide. Cyanobacteria undergo oxygenic photosynthesis, and we're going to see they do have chlorophyll, the same chlorophyll A that we find in plants. Other phototrophic bacteria generally have bacterial chlorophylls, so a different version. But both can have carotenoids. So carotenoids are pigments that are also found in plants, but also found in phototrophic bacteria. Most phototrophic bacteria are going to be classified by their color, so what color they appear, as well as whether or not they use some kind of sulfur compound as their electron donor. So we'll see the names include purple, green, or sulfur versus non-sulfur groups. For sulfur groups, they are going to be using hydrogen sulfide or something similar as their electron donor. For this section, you don't have to worry about the names that I've provided here. I just wanted to give you examples from our textbook. But look at the general categories shown here. You can see they're categorized based on their color, so what color they appear based on the pigments they have, and whether they use sulfur compounds or non-sulfur compounds or molecules as their electron donor. So purple sulfur bacteria, for example, uses hydrogen sulfide. The only group where I do want you to know their specific name are those cyanobacteria, because they undergo oxygenic photosynthesis. And they can also fix nitrogen in cells that are called heterocysts. So we're going to see cyanobacteria form these filament-like structures that are multicellular, actually, when they form these filaments. And in that filament, you'll see cells known as heterocysts that go through nitrogen fixation. Our book gives us some pictures of these purple and green bacteria. The purple is a little bit hard to see, but we can definitely see the green here. Cyanobacteria can be found in many different types of habitats, including in the water, in soil, and they can even be found on rocks. So the book mentions how scientists found cyanobacteria in rocks in Antarctica, where the temperature can get to minus 15 degrees Celsius, and there's barely even any moisture or rain there. So they found this very interesting because this environment in Antarctica really was very similar to those on Mars. So maybe cyanobacteria could grow there, could grow on Mars. So remember, these are bacteria that go through oxygenic photosynthesis. They produce oxygen when they undergo photosynthesis, and they also have a plant pigment, or a pigment also found in plants called chlorophyll A. So that gives them their green color. But if you look at cyanobacteria, they actually look kind of bluish green. And this is because they have additional secondary pigments like phycocyanin and cyanophycin. So we think that Cyanobacteria were the precursors to what plant chloroplasts are today through endosymbiosis theory. Remember, this is when a larger eukaryotic cell engulfed a prokaryotic cell, perhaps the cyanobacterium, and over time the cyanobacteria evolved and became those chloroplasts. That would explain why chloroplasts have their own DNA, but it's prokaryotic DNA, as well as their prokaryotic ribosomes. There are some genera of cyanobacteria that can be harmful. For example, Microcystis can form toxic blooms. So when these cyanobacteria overgrow, they can release toxins that can be harmful to both animals and humans. Here on the left, we see an example of cyanobacterium, specifically Microcystis aeruginosa. This is found in freshwater environments. On the right, we see an example of a toxic bloom. So this is when the cyanobacteria overgrow. This often happens when we have warmer temperatures. And it's hard to tell the difference. I can't just tell by looking at this picture if it's an algal bloom or the bloom of these bacteria. You could take a water sample and look at it under the microscope or perform additional metabolic tests. I wanted to show you also that these cyanobacteria form these filament-like structures, right? These string-like structures. If you zoom in, you can see individual cells, as well as those heterocysts where nitrogen fixation occurs. Here's the table from our book summarizing phototrophic bacteria. Notice that cyanobacterium is listed here. I want you to know Microcystis, the genus name, and that these are oxygenic. The common name for these bacteria are blue-green bacteria, remember, named after their color. We also saw these previously, bacteria named after their color, as well as whether or not they use sulfur, some kind of sulfur compound, as their electron donor. The rest of these are anoxygenic. They do not produce oxygen through photosynthesis. And notice that there are both proteobacteria and non-proteobacteria. Don't worry about the genus names listed here. For our phototrophic bacteria checkpoint, remember that phototrophic bacteria are different from other prokaryotes because they use sunlight as their primary energy source, right? They can go through photosynthesis. And these bacteria have specialized pigments like bacterial chlorophylls and carotenoids that allow them to capture light energy, and they can perform either oxygenic photosynthesis or anoxygenic photosynthesis. Oxygenic is when you produce oxygen, anoxygenic is when you don't. And remember that the cyanobacteria actually have chlorophyll A similar to plants. All right, let's recap section 4.3. Looking at unique features of non-proteobacteria, gram-negative bacteria. These are not part of the Proteobacteria phylum, but they're still gram-negative. We had pretty diverse groups: chlamydia, spirochetes, the CFB group, and planktomycetes. These were pretty adapted to specialized environments. We had examples of intracellular pathogens like chlamydia. Spirochetes had spiral motility. The CFB group included anaerobic fermenters, and planktomycetes could bud, so their reproductive process was through budding. For examples, I just kind of mentioned them, but we had chlamydia, spirochetes, CFB group, and the planktomycetes. For the third one, unique features of phototrophic bacteria. These use sunlight as their energy source. They can go through photosynthesis. They had pigments like bacterial chlorophylls or carotenoids. They could be oxygenic or anoxygenic, and they are often classified by color and also by sulfur versus non-sulfur metabolism. Examples of these, we saw things like purple sulfur bacteria, green sulfur bacteria, etc. But cyanobacteria is probably the most common one that you'll encounter, such as Microcystis aeruginosa, where we saw that picture of the toxic bloom in what it looked like a lake. That takes us to the end of part 3. In our fourth video, we're going to switch gears and start looking at gram-positive bacteria.